Dear students, next topic is Quantitative Trait Loci or QTL mapping.
Before going to the topic, please subscribe my channel. Press the bell icon for notifications. Like, share among your friends and comment.
The loci controlling quantitative traits are called Quantitative Trait Loci or QTL. The term was first coined by Gelderman in 1975. It is a region of the genome that is associated with an effect on a quantitative trait. It can be a single gene or cluster of linked genes that affect the trait. Next, we can see the characteristics of QTLs. These traits are controlled by multiple genes, each segregating according to Mendel's law. These traits can also be affected by the environment to varying degrees. Many genes control any given trait and allelic variations are fully functional. Individual gene effects is small and the genes involved can be dominant or codominant. The genes involved can be subject to epistasis or pleiotropic effect. Then what is QTL mapping? The process of constructing linkage maps and conducting QTL analysis, that is to identify genomic regions associated with the trait, is known as QTL mapping. Identification and location of polygenes or QTL by use of DNA markers. It involves testing DNA markers throughout the genome for the likelihood that they are associated with a QTL. The principle: genes and markers segregate via chromosome recombination during meiosis, thus allowing their analysis in the progeny. The detection of association between phenotype and genotype of markers. QTL analysis depends on the linkage disequilibrium. QTL analysis is usually undertaken in segregating mapping populations. Then what are the requirements for QTL mapping? Availability of a good linkage map, a segregating population, a good assay for the traits of interest, software available for the analysis, molecular markers, and sophisticated laboratory. Next, we can see the steps involved in QTL mapping. First one, selection of parental lines. That should be sufficient polymorphism. Parental lines are highly contrasting phenotypically, then should be genetically divergent. Second is selection of molecular markers, dominant or codominant, making crosses, creation of mapping population. Here is a summary of QTL analysis. There are two parents selected, recombinant inbred lines. Then genotype with molecular markers were added. Then analyze trait data for each line. Link trait data with marker data. Mapping run with the mapping software. Trait QTL mapped at bottom of small chromosome. It's not easy to do this analysis manually, and so with the help of a computer and a software it is done. The segregation data of both the phenotype and the genotype are collected and arranged in an Excel sheet for QTL analysis using the appropriate software. See, this is one of the free model software, and here we can do interval mapping, regression, QTL presence and estimate position in a map, simple interval mapping, a single QTL without effects, composite interval mapping, a single QTL with effects on other QTL. Here is the merits of QTL mapping. First one is identification of novel genes. Where mutant approaches fail to detect genes with phenotypic function, QTL mapping can help. Good alternative when mutant screening is laborious and expensive. Example, circadian rhythm screens can identify new functional alleles of non-function gene. Example, flowering time QTL, EDA was the Cry2 gene. Natural variation studies provide insight into the origin of plant evolution. As an example of QTL analysis, we can consider the phenotypic trait in Drosophila. The fact that traits with continuous variation can be controlled by genes dispersed over the whole genome was shown in 1957 by James Crow. In 1916, he studied DDT resistant in Drosophila. A DDT resistant strain of flies was created by growing them on increasing concentration of the insecticide. Crow then systematically tested each chromosome for the amount of resistance it conferred. Susceptible flies were mated with resistant flies, and the male from these crows were backcrossed. Offspring were then scored for the particular resistant chromosome they contained and were tested for their resistance to DDT by adding markers. Males were used in the backrows because there is no crossing over in males. Therefore, resistant and susceptible chromosome were passed on intact by the males. The results are shown in the figure. The results indicate that each chromosomes in Drosophila contain genes that contribute to DDT resistance. In other words, the loci bearing the genes that control DDT resistance are scattered throughout the genome. Dear friends, thank you for watching. Don't forget to leave a like and please subscribe my channel.